my pleasure to introduce our speaker, Dr. Heather Bryan, who is currently a professor at the University of Northern British Columbia in Canada. She did her undergrad studies at the University of Victoria, which is in British Columbia. She moved east just a little bit, one province over to Alberta, where she did her PhD studies at the University of Calgary. And it was during her time or shortly thereafter that we first interacted. I remember handling one of Heather's papers on bears. I think it was probably the first bear paper I'd handled. I'm a fish researcher for anybody that doesn't know. And since then, there have been an awful lot of bear papers, and Heather has certainly helped us on many occasions as a referee and contributor. So Dr. Bryan then headed back west to University of Victoria, where she held a number of postdoctoral positions before joining the professoriate. The title of her presentation, I think, does a really nice job of summing up the body of work and themes that she tackles in her research. And so I'm not going to restate that here. It's on the slide, but it's fascinating work at the intersection of predators and prey. And so I'm sure we're all going to gain a lot from this presentation.
Well, thank you everyone for joining today. I'm joining you today from my office at the University of Northern British Columbia, which is located on the unceded traditional territory of the Lheidli-T'enneh Nation. It's a beautiful fall morning here today, about just past 8 o'clock Pacific time. But today, at first, I'd like to take you about 500 kilometers northwest of where I am today to this scene that I have here on the screen of a caribou swimming through the water. And so this picture was taken in Tahltan territory. And at the time I was on a canoe trip in the headwaters of the Stikine River. And my family and I were camping on the side of the lake. And we woke up to this amazing scene of a figure, this caribou emerging from the mist and swimming across the lake and popping out just beside our campsite, looking more surprised to see us than we were to see it. And then proceeding into the nearby bushes. Just seconds after that, we were treated to another site, which was this moose also emerging through the mist, swimming across the lake, popping out beside our campsite and moving into the nearby bushes. And it was this experience that I thought of when I was putting together this talk, because it reminded me of the incredible adaptations that wildlife have on a day-to-day basis for coping with predictable variability in their environment, like predators, as well as more unpredictable events like coming across campers in their somewhere they weren't expecting them to be. And what we assumed happened on this morning is that probably there was a predator or some other disturbance that motivated both of these animals to jump into the water, swim a couple kilometers, and come out on the other side. And so what I'm really interested in my research is understanding how wildlife respond to kind of predictable, common types of disturbance that they encounter on a day-to-day basis, as well as potentially longer-term stressors related to changing and less predictable types of environmental change. And as we know today, in today's world, wildlife around the globe are facing an increasing number of changes in their environment, whether it's increasing severity and frequency of wildfires, unprecedented amounts of landscape change through harvest practices, as well as climate change and many more things. And so I find the field of conservation physiology a really valuable lens for trying to understand the responses between what's happening at the individual level, how organisms respond to external stressors and stimuli. I'm really grateful to Dr. Cook and others who worked very hard to formalize this discipline and to develop theory and a framework for this type of work that can be applied broadly to many different ecosystems, including through the formalization of the journal Conservation Physiology. And today I'd like to focus on a few examples where I consider that we've used this lens of conservation physiology to try and understand effects of environmental change on different species. So I'm going to be taking you on a bit of a whirlwind tour of different ecosystems and predator-prey systems that are found in different parts of British Columbia. And all are united in that these species are all iconic and connected to people through their importance culturally, through the resources shared by both wildlife and people, and through the changing environment that all of us share. So I'll start by talking about bear salmon systems, and then which is based on some past work that I've done, and then I'll move into talking about some of the more recent work that my lab group is focusing on in various ungulate systems. So bears are an iconic predator of salmon that have co-evolved with abundant returns of spawning salmon for thousands of years. The population health of bears is so closely connected to salmon. Bears that have access to salmon are larger in body size, they have more babies, so their litters are larger, and they exist at higher population densities. Salmon can also structure social dynamics in bears, where bears can form dominance hierarchies around accessing salmon. So this map shows the range of grizzly bears past and present. And so if you look in kind of the recent past, grizzly bears have been extirpated from much of the southern United States. They still occur in much of Canada and Alaska. But throughout a lot of their habitat, they have experienced a lot of habitat loss and fragmentation. There's one part of coastal British Columbia, so this region here, hopefully people can see my pointer here. This area that spans from the northern tip of Vancouver Island, this is on the west coast of British Columbia, north to the southeast corner of Alaska here. This is known as the Great Bear Rainforest. It's an area of immense beauty. It's largely roadless. And it's a place where grizzly bears, which are also known as brown bears in other parts of the world, we call them grizzly bears mostly in coastal British Columbia. These grizzly bears coexist with other predators, including black bears, which have a rare white morph in their coat color in some parts of coastal BC. And so you get the white morph or the spirit bear as well. There's also wolves that coexist with bears in this area. And since time before memory, these large carnivores have coexisted with the First Nations who live in the area and who have profound impact shaping and caring for the lands and waters. And for many nations, the large carnivores are culturally and spiritually important, and they feature prominently in the traditional stories, dances, and knowledge. people and carnivores are connected in part through spawning salmon. These return in huge numbers to coastal waterways and provide a rich source of food for carnivores and people alike. But today, the relationship between salmon and people has changed through industrial fishing practices. In some of the fisheries, people take 50% of fish returning to spawn every year. In some cases, even higher proportions each year are taken. And that means that the remaining fish that escape the fishing nets have to be shared among many different consumers of salmon, including bears, but also many other species of invertebrates and vertebrates also feast on the salmon returns each fall. So emerging in response to widespread salmon declines in coastal BC, as well as other threats to bear health, has been a long-term monitoring program. So this monitoring program is a partnership among 5 coastal First Nations, including the Newhalk, the Wikinew, Haislik, Gitgat, and Kitasoo-Hey-Hey nations. And the monitoring program actually began in 2006, was led by William Housey, who's a health-sick scientist and cultural leader who lives in Bella Bella, this community here. And since William started that study, the project has expanded out to an area that's over 23,000 square kilometers. And there's been data that has been collected, again, almost for 15 years or more now. Each one of these little dots on the map represents a site where people monitor bears using non-invasive methods, so barbed wire hair snags that are placed in the forest. And these hair snags of barbed wire is used to collect these little tufts of hair and inform many different long-term research questions. These pictures here on the side are some of the many people who have been involved in this work over the years. It takes a small army of people to do this work, and it's really a labor of love, and a lot of people have put a lot of effort into this ongoing project. One of the questions that we asked early on in this work was how bears might respond physiologically to salmon declines. And this was formed the basis of a lot of part of the graduate work that I did. And so we reasoned that one mechanism might be that bears, if they're not getting enough salmon, they might experience nutritional stress due to not having enough of this really important resource. But we also predicted that there might be some social consequences associated with low salmon returns, and that might alter the social dynamics among bears that feed on this very spatially and temporally constrained resource. And so this picture is from McNeil Falls in Alaska. It's not from coastal BC, but it's such a great picture that shows all these bears foraging in the same area. That is why I like to use it. And so to kind of look at these different hypotheses and try to figure out which whether nutritional stress or social stress might be most likely to influence salmon bear related to salmon bear interactions, we looked at hormones in hair samples. And so we looked at cortisol which is a kind of a general reflection of physiological processes, including nutrition. But cortisol can also be modulated by social interactions. So we thought this marker might represent either nutritional or the social competitive environment. We also looked at a second hormone in hair, which was testosterone. And drawing on kind of bird literature, and particularly work by John Wingfield on the challenge hypothesis, we reason that perhaps it may also be related to the social competitive environment in bears. So so what we did is we put out these barbed wire fences in the forest. I'm not sure if you can see it very well, but there is a barbed wire behind this bear. And we add what's called a non-reward bait. So bears have an amazing sense of smell, and they will come and investigate anything new and interesting in their environment. So we put out dogfish oil and leave the bait out there for about 10 days. Bears come visit the site, and if we're lucky, they leave behind a little tuft of their hair on the barbed wire. This map shows different sites where we detected grizzly bears. And again, this was early on in this larger study. There's now many, many more samples from bears, but we were able to sample about, I think, 27 bears initially. And we augmented our sample size using hair collected from hunted bears in the same area. So even though we only had a few milligrams, so 20 to 50 milligrams of hair from these bears, we got as much information as we could out of those samples. So imagine we have a hair sample here. We collected, we used DNA, which is generally most concentrated in the follicles of the hair. And that was used to identify the sex and the species of the bear. And then we used the hair shafts for stable isotope analysis, and that allowed us to look at the diet, the proportion of salmon versus plants in that a bear had been consuming. And then we also measured cortisol and testosterone in the hair samples using enzyme immunoassays. And so I'll just go through what I think are kind of the most interesting results from this work. And so this figure shows cortisol here on the left y-axis, and then the hair growth here on the y. I'll talk about the salmon in a second. So we had measurements of cortisol in hair in 2008, 2009, and 2010. And so the interesting thing here is that there was kind of lower cortisol levels in the hair in 2010 compared with 2009. We then looked at the salmon consumption by bears in also using the hair samples. And there's kind of an interesting lag where 2008 was a very, very low year for salmon on the coast. And so the salmon levels here in the blue on the left there were quite low in the hair. The year after that low year, the bears had higher cortisol. 2009 was a really good salmon year. And so the year after that, the cortisol in the hair of those bears was lower. So we had a small sample size for this, but really it fits with our understanding of bear physiology and the importance of salmon, where bears going into hibernation in the fall that are in better nutritional condition probably come out of hibernation in better condition as well, and then they have lower cortisol in their hair the following year. So this is something I'd like to follow up on with more now that there's more samples and more data, but it does fit with kind of understanding of the seasonal importance of salmon to bears. So that was looking at the amount of salmon bears were actually eating using those stable isotope signatures in the hair. But the amount of salmon bears actually eat only represents one component of that resource. And the other component that's important to consider is how much salmon are actually available. Because that can influence how many bears in a population might gain access to those salmon. And so to get at availability, we use data from the Department of Fisheries and Oceans on salmon escapement. And we measured the amount of salmon bears would have in their typical home range. So for these bears, we didn't have colors on them, so we don't have actual estimates of the home range of individual bears, but we assume that bears have a kind of a standard home range that where they would maximize their access to salmon. And then we calculated roughly how much salmon was available to each bear using those assumptions. And the most interesting finding, I think, that came out of this was some relationships between salmon abundance and testosterone levels. And so this is a figure on grizzly bears that consumed either low or high amounts of salmon. And on the y-axis is the testosterone level in the hair. So here, those bears that didn't eat a lot of salmon showed similar levels of testosterone in their hair when salmon availability was low versus when it was high. So it didn't really change much. The interesting thing occurred in bears that did consume a lot of salmon. So for bears here, the testosterone levels were lower when salmon was more abundant on the landscape. And so one explanation for this is that when there's a lot of salmon, the social competitive environment is more relaxed. There's less need to vie for access to salmon, and perhaps that's why bears showed lower testosterone. There's other explanations as well, such as possible effects of salmon consumption on testosterone levels, but we've had some other findings that I think were consistent with the hypothesis around the social competitive environment. So just to kind of wrap up this part, and then I'll maybe quickly touch on a couple of other studies that we're working on now. Is that what we inferred from this work is that there may be ways by which salmon declines could influence both nutritional and social processes in bears. And of course, the variability that we saw is likely reflective of adaptive responses that bears have to environmental variability, including in their food resources. But what might happen under extreme food shortages is that lead to chronic stress is there could potentially be fitness consequences. There's some also interesting theoretical implications, and I love talking about social modulation of testosterone. So if anyone's interested in following up, that would be great, because I think it's a very interesting line of research. There's also some interesting management implications from this work in terms of the importance of ensuring that there's enough fish that return to spawn to provide for bears as well as for human needs. And actually, the Wikinew Nation has done some really amazing work about ways of incorporating the needs of other species, including bears, into their fisheries management practices, which is really novel and exciting. And then another example is that kind of understanding the ways by which salmon abundance affects bear stress levels and possibly testosterone provides emphasizes the importance of minimizing disturbances for bears that are feeding on salmon. Okay, so I know we don't have a lot of time. I'll just briefly touch on a few other projects that are going on in our lab that where we're using similar approaches. And I'll go first to move from coastal forests to interior forests. And in interior forests of BC, moose are the salmon in these forests. They are culturally important to many First Nations. They're an important prey species, not only for people, but also for predators like wolves. And they've been very affected by recent changes in forest harvest practices. So back in the early 2000s in Canada, in BC, we experienced an epidemic of pine beetle that killed a lot of the forests across the province. And following that pine beetle epidemic, there was a move to remove those dead trees before they fell down or created forest fires or became unmarketable. And that precipitated massive amounts of salvage logging. So around that time of the salvage logging, there was also changes in moose populations, where some moose populations declined by up to 70% in the province. So the provincial government has initiated this large research program that's been going on for 10 years now, where they've been investigating underlying drivers of these declines in 5 study areas. So this map shows the 5 different study areas. The provincial biologists have collared more than 500 moose and have also collected a lot of health-related data as part of these captures. So as an academic partner with the province, I'm working with government biologists, other academics, and graduate students to try and figure out some of the underlying mechanisms that might be driving the declines. So here, this is one of the graduate students that I work with, Carl Jeffries. He's looking at understanding, trying to better understand the mechanisms of why some of the moose in this study have been dying of what's called apparent starvation, which is poor nutrition that doesn't have kind of other causes and other kind of nothing else is wrong with some of the moose that are dying other than them being in poor condition. And so there's been some work to find out that it's really the moose that use early cereal clear cuts that are more likely to die compared with moose that are using older forest stands. There's also some work showing that the nutritional quality of forage in these clear cuts may not be high enough to support moose. But we don't know for sure kind of what's going on. And so Carl's trying to figure out, test different hypotheses, whether this poor nutrition could be related to thermal stress, nutritional quality, or altered predation risk in these habitats. And so he's using hair cortisol and body fat measurements. He's also collecting additional data. He's following some of the collared moose to collect urine samples and pellet samples to look at some of the physiological responses to weather events and snow conditions in the winter. And so here he is digging out his snowmobile. He's spent a lot of time digging out, trying to get unstuck out of the snow as he was following these moose in less than ideal conditions to collect their urine and their pellets. So in moving a little bit further north from the moose work, about 2 hours north of here in Prince George, is a herd of caribou called the Kennedy siding herd. They're a herd of central mountain caribou. And And about 10 years ago, their population numbers declined to less than 50. So there was serious concerns around their survival of the herd. And so the McLeod Lake Indian Band, along with Doug Hurd of Titanus Wildlife Research, initiated a supplemental feeding program as an emergency method to try and stop the declines. And so this supplemental feeding program has been implemented now for 8 years, and it's been highly successful. The population is now up at 114 animals. And so where I'm working with the graduate student, Lauren Elvis, and what we're trying to do is trying to understand the link between that population change and supplemental feeding using physiological bioindicators. And this is a really neat setup because these caribou come into the feeders. It's possible to identify every single individual using their antler characteristics and other features. Here's a few examples of caribou that come that Lauren and Doug have identified. These animals, so this means that we can collect samples of pellets and hair from known individuals and collect them repeatedly over the period when they're at the feeders. We've also have a scale, a body weight scale set up with a salt lick here. So the caribou come onto the scale while they're at their feeders to get the salt. And there's a remote camera that is set up to take weight measurements and height measurements of the caribou. So we can really track individual condition over time. And so what Lauren is doing for her graduate work is looking at hormone levels and hair samples and pellets to try and get at whether this population boost is coming from increased female pregnancy or rather increased calf survival. So I won't go into a lot of details there, but happy to talk about that more later. And I'm almost out of time. I'm just going to briefly mention one final example here. And we're just going to move even further north here to an area of northern British Columbia called the Musqueam Management Area. This area is also known as the Serengeti of the North. It's a place where many species of ungulates, including the ones I've got here on the slide, live at fairly high numbers relative to elsewhere, and hence the name that this area has gotten comparable to the Serengeti. So this region is massive. It's the size of Ireland. It lies on the traditional territories of the Cascadenai, the Treaty 8, and the Carriers-Sicani nations. And it's largely roadless. So the only road that goes through is in this northeastern corner, which is the Alaska Highway. So it's a pretty special place in terms of the cultural history and the biomass. One of the management practices that has been used in this area for millennia by First Nations people, and more recently by guide outfitters and provincial government biologists, has been prescribed burns. So prescribed burns are used to promote grassland habitat, which is good for ungulate species, and has probably been part of maintaining this area as a place that with very high ungulate abundance. But in the last 10 years, there's been policy changes where fires haven't been implemented as often. And so that's led to habitat changes where there's been conversion of shifts in grassland towards shrubs and more aspen dominated areas. There's also changes with climate change and with the increasing tree line coming up that is also encroaching on grassland. So what colleagues and I are doing from the, so Alicia, I'm working with Alicia Woods of Ridgeline Wildlife Enhancement and as well as the Wild Sheep Society on this project. And we're implementing prescribed burns using before-after control impact design, and we're looking at changes in vegetation as well as responses in the sheep to the prescribed burns. So this slide has a few pictures from the fieldwork. We're doing recruitment surveys to try and look at the link between the burns and population recruitment. We're collecting pellet samples in different times of the year to look at physiological changes in hormone levels, but also nitrogen content of the food and various other things. And then we're monitoring these sites by cameras. And so this is Alicia and Jody. They're trying to put up a camera where there's no trees. So they've strapped it to a rock here. So you have to get pretty creative in some of these high elevation sites. And then here's some of the camera images of sheep that are foraging in some of these subalpine areas. So I think I'll stop there so that there's time for questions. I think I think just to conclude, I'm really grateful again to have the lens of conservation physiology that's been now nicely defined as a field. I think it's a really important framework for going forward. And I'm really excited about kind of the future areas and findings that we'll get using these approaches. I'd like to thank lab members. Here's some of our lab group here in the photo. I'd like to thank all of them for their hard work and all my partners and funders.